Welcome, everyone. This is our last McMaster speaker for our lecture series. Joining us today, we have Dr. Giuseppe Melanchini. I hope I'm pronouncing that correctly. Dr. Melanchini is a professor at McMaster University. His research focuses mainly on signaling, kinases, and intrinsically disordered proteins. Kinases and intrinsically disordered proteins play a critical role in cancer, and hopefully we'll get a thorough explanation as to the function of these during the presentation today. Any questions that you might have, I'm sure Dr. Melanchini will be excited for you to unmute yourself and ask the question with your voice, but if you feel uncomfortable, that's completely fine. You could drop it in the chat and I'll read it, whether it's at the end of the lecture or depending on how Dr. Melanchini feels.
Thank you so much, Israel. Can you guys hear me well? Wonderful. Yeah, so you please feel free to interrupt me anytime if you have questions. If you prefer to ask at the end, that's fine. If you prefer to ask questions as we go along, that's fine too. I just want this to be as useful to you and the audience in general as possible. So please don't hesitate to ask questions. And I'm sure if you ask a question, other people will probably have similar questions, including those who cannot be here right now. So again, don't hesitate. I would like to start by thanking Israel for organizing this wonderful initiative. And also, I would like to thank my collaborators in the lab who actually did most of the work that I'm going to present today, in particular, Dr. Akimoto, Dr. Bune, Dr. Van Schouwen, Dr. Bolton, and Dr. Defari. As you see, they're all doctors because eventually they defended their PhD or they joined my lab with a PhD from somewhere else as postdoc. But at one point, they were undergraduate students, and many of them here in Ontario like you. So this can also give you an idea of the graduate research that you can do after you earn your bachelor degree in biomolecular biological sciences. So I just start with a brief outline of what you can expect in this hour or less that we're going to spend together. So first, I'll try to explain why we bother actually trying to understand the mechanism of action, or MOA, of allosteric kinase inhibitors. Why do we care? After that, I will try to understand what controls the vertical scaling of a typical dose response curve. So the dose, for example, could be the concentration of an allosteric kinase inhibitor that is administered to a living system, and the response could be the kinase activity of a given specific kinase. So we'll try to understand what modulates the response at plateau, at saturation of this dose response curve. Now,
Dr. Melanchini, your screen is not shared if you're sharing your screen currently.
Oh, it's not shared?
No.
Oh. Okay, thank you for letting me know. So then I should actually go back. Just a second. Okay. Sorry about this. I had it shared before and then probably went away. Okay. It should be shared now. Can you hear me?
Yep, everything's good now.
Oh, okay, great. So, well, I'll go backwards a little bit very quickly. So you heard me talking about my collaborators and how grateful I am to them, and they were undergraduate students in Ontario like you, and these are their pictures. I already mentioned their names. And I was just trying to give you a brief outline of the talk. First, I will try to explain why we care about understanding the mechanism of action, or MOA, of allosteric kinase inhibitors. And then I'll try to understand what determine the vertical scaling, so the response of a given kinase at a given dose in a typical dose response curve for allosteric kinase inhibitors. But this only tells us how well the kinase responds to this inhibitor at a given dose of saturation. So it's important to understand what determines the horizontal scaling of these dose response curves, because the lower is the concentration needed to obtain the desired inhibition, the lower is the chance that we have unwanted side effects, okay? So these would be the three main pillars of the talk. Any questions so far? I cannot see you, so just unless you respond, I don't have any sign of feedback.
Yeah, everything's
Okay,
good so far.
Wonderful. So why do we care about allosteric kinase inhibitors? Well, primary reason is because kinase inhibition has a huge clinical potential for a multitude of diseases, from infectious diseases such as malaria to antibiotic resistance. It's also very promising as a treatment of cancer, especially leukemia. And there are actually allosteric inhibitors that are now commonly used in the clinical practice. Also has a lot of potential for the treatment of neurodegenerative diseases that are often triggered by the hyperphosphorylation of intrinsically disordered proteins. So essentially, you can pick key disease or disorder, and it's often possible to find a kinase associated with it that, when inhibited, elicit a desirable therapeutic outcome. And usually you identify kinases in biological jargon because they are indicated by K. So if you have an abbreviation with a K, the K usually stands for a kinase. For example, here for malaria, we target the Plasmodium falciparum protein kinase G. So K is for kinase, and that's exactly GMP-dependent kinase. Now, in terms of biochemistry, we all know that kinases are essentially enzymes, so they are catalysts of a very important reaction that leads to post-translational modifications of a protein. And this reaction is actually the phosphoryl transfer from ATP to serine, threonine, or tyrosine. So all amino acids that have an OH and hydroxyl group in their side chain that gets phosphorylated. And when it gets phosphorylated, often that triggers either conformational changes or dynamical changes or interactions with other proteins that actually regulate downstream signaling pathways and eventually the whole metabolism of the cell. So they are a very important regulator of cell metabolism, and that's where this clinical potential comes from. However, when there is also an opportunity, there is also a challenge. And the challenge in kinase inhibition is that although we do have many different kinases for many different diseases, they are all quite similar when it comes to their active site. And that's actually quite expected if you think about it from an evolutionary perspective, because all these enzymes have evolved to catalyze the same reaction. As I said before, the transfer of a phosphate group from ATP, or magnesium ATP more precisely, to the OH, to the hydroxyl group of serine, threonine, and tyrosine of substrate proteins. So that's why the different kinases all have very similar active sites. So if you were to target these kinases, if you were to try to inhibit these kinases simply by focusing exclusively on the active site, then it would be actually quite challenging and difficult to be able to find a selective kinase inhibitor. You will probably find an effective kinase inhibitor that is very good at inhibiting a given kinase. For example, the Plasmodium falciparum kinase, but that same inhibitor will probably inhibit kinases that are also in the human host of a patient with malaria. So you won't have the pathogen versus human host selectivity that that is desirable. So how do we solve this selectivity problem? Well, one promising avenue is that rather than targeting the active site that is so conserved across different kinases, we target what we call allosteric sites. So allosteric sites are different from the active site. So they don't have a direct role in the enzymatic function of kinases. So they don't necessarily catalyze the phosphorylation transfer from magnesium ATP to substrate. However, they are able to regulate what happens at the level of the active site. Since they don't have a direct role, they don't have a direct enzymatic role, they're not subject to that evolutionary pressure for conservation. And therefore, often happens that we could have two kinases with fairly similar active site, but different allosteric sites. So we can target these allosteric sites, and these allosteric sites are still able to regulate what happens in an indirect manner, what happens in the active site. So you not only have the advantage of engineering more selectivity in your inhibition of the kinase, but also you have the advantage that you don't have to compete with the endogenous substrates, which always requires higher concentrations of inhibitors, and that, as we said before, increases the chances of undesired side effects. Okay, so that's why we're interested in allosteric inhibitors. But as we said before, there is always a catch. Where there are opportunities, there are challenges, and vice versa. So here we have an opportunity of selective allosteric inhibition, but the challenge is that allosteric inhibition is quite complex. What do I mean by this complexity? Well, if you look at the classical understanding of allostery, usually the picture goes as follows. You pick a kinase, the kinase has an active site and a given allosteric site. You do wonderful structure-based or screening to find an allosteric ligand that target that active site. And then you assume that the response of that given kinase to the given ligand at that given allosteric site is predetermined. So it's either agonist, so as you increase the concentration of the allosteric modulator, the kinase activity goes up compared to baseline, or as you increase the concentration of the allosteric modulator, the kinase stays inhibited, and that's what we call antagonism. And according to the classical view of allostery, we have either or response. So we have either an agonistic response or antagonistic response. But once you define the allosteric pocket and the allosteric ligand for a given kinase, you cannot have a switch between the two. Now, this classical view of allostery is now being challenged because we are coming to appreciate several counterexamples that the switches between agonism and antagonism are actually possible, even without changing the allosteric binding site, even without changing the allosteric ligand, even without changing the kinase and how the two interact. Because what drives often this switch between agonistic and antagonistic responses to allosteric modulator is actually the subcellular, metabolic, and proteomic context. But how the local context of the cell actually influences the agonist versus the antagonist is actually not very well understood. We know that this happens from multiple observations that I'm going to present now, but the mechanism of how this happens at the molecular level is actually at the forefront of allosteric research, and I'll show you later the first step in this direction. So one of the first examples in which we came to appreciate the presence of allosteric pluripotency was actually from clinical practice. For example, tamoxifen is known to work very well as an estrogen receptor inhibitor in breast tissue, but in the uterine tissue, actually, it activates estrogen receptors. So two very different responses for the same receptor, but in different tissues and presumably in different local environment. So this was the first clinical evidence that was a warning and an eye-opener towards allosteric pluripotency. But we can find other examples of allosteric pluripotency also for very famous prototypical kinases that you may have already encountered in your studies of cellular biology and metabolism. For example, protein kinase A is also called the cyclic MP-dependent protein kinase. As you can appreciate from this figure, is a major signaling hub. Historically, it's also served as a prototype for the kinase superfamily in the sense that the structures of protein kinase A have been solved before other kinases, probably because the biochemistry and the expression and purification of this protein was more established, primarily because it's composed of two different subunits that can be expressed and purified separately. And it's also important in medical terms because several mutations in pKa are related to different diseases. If pKa is overactivated, we have what is called the Carney complex, which is a predisposition towards many different forms of cancer. Or if pKa is not responding well enough, we have a form of hormonal resistance that leads to developmental disorders or pediatric disorders that are commonly referred to as acrodysostoses. So, but we know that pKa is very important in the cardiovascular field as well. And to understand that, I show you here like a simplified signaling scheme. So, for example, you have epinephrine, so an extracellular signal that stimulates a G protein couple receptor or GPCR. This in turn, through the G protein, will activate adenylyl cyclases, so AC. And then the increase of cyclic MP will activate among many different proteins, protein kinase A. Now, protein kinase A in turn controls calcium metabolism, which in turn regulates how fast and how strongly the heart beats. So a very promising strategy to decrease blood pressure is actually relying on what are called beta blockers. So beta blockers act on a class of GPCRs called beta adrenergic receptors. So essentially, they don't allow epinephrine to be to activate effectively the GPCR. And then the net result is that they tone down cyclic MP signaling and especially the activation of pKa. And that basically makes the heart beat not as fast, not as strongly, and it decreases the flow of blood in the arteries, and that decreases the blood pressure. But the problem of these strategies, however effective are the beta blockers, is that you are trying to manipulate these signaling pathways very much upstream. So you're not being that selective because you're affecting the whole cyclic MP dependent signaling pathways in the cell. So not only pKa signaling, but also other forms of signaling, like, for example, EPAC, exchange protein directly activated by cyclic MP, and other IO channels that are gated by cyclic MP. So ideally, you would like to take advantage of this idea of inhibiting pKa to decrease blood pressure, but without having to interfere with all the other receptors for cyclic MP. You would have the same problem if you try to inhibit what we call phosphodiesterases, called PDE here, because phosphodiesterases essentially try to stop the cyclic MP signal by hydrolyzing cyclic AMP, so the cyclic adenosine monophosphate into 5' AMP. So they open the phosphodiester bond. And once they open that phosphodiester bond, then AMP is not really able to effectively bind and activate pKa. So that's how you stop the signaling, but you would have the same problem. You would have a non-selective interference from EPAC and HCN as well. So we are interested in a more selective approach in line of what I also explained before about allosteric inhibition. And we would like to target more directly pKa using cyclic AMP analogs that are actually selective for pKa. So let's try to understand how cyclic AMP works. But before I do that, let me ask you if you have any question about what we saw so far. Is this clear?
Yep. Maybe someone in the chat might have a question. Oh, seems
Yes,
like
so
no
clear.
one understands it.
Thank you very much. Sorry, I have to remove the pointer to move the chat box and then go back here. You can still see my pointer quite clearly, or no?
Yeah.
You can see it?
Yes. Yeah.
Okay. Okay, great. So we said we wanted to understand how cyclic AMP works. So again, that's something that you may have seen in textbooks, but you know that under resting conditions, pKa exists in an inhibited state in which the regulatory subunit, which is shown here in red. I'm not showing the whole regulatory subunit for the sake of simplicity, but just the part that is really important for inhibition, is actually bound to the catalytic subunit of pKa. So the catalytic subunit shown here in gray is the actual enzyme that catalyzes the phosphoryl transfer. Now, it's important to keep in mind that this is a very stable complex in the presence of magnesium ATP that acts like a glue that keeps the regulatory subunit shown here in red together bound with the catalytic subunit shown here in gray. And the same structure is shown schematically here as a cartoon on the right. These cartoons are a simplified picture of the high resolution 3D structures, but cartoons are actually useful later on when we try to see the interplay between all these different structures. So it's important to understand that magnesium ATP stabilizes the interaction between the regulatory and the catalytic subunit because it really anchors this linker shown here of the regulatory subunit into the active site of pKa. Now, what happens to this inhibitory complex when cyclic AMP binds? Well, cyclic AMP binds to both domains in the regulatory subunit indicated here by A and B, and it causes a conformational switch in both domains. The conformational switch in both domains has two major effects. First, it brings them closer to each other. And second, it weakens the interaction with the cis subunit. So to cut a long story short, once the regulatory subunit binds a cyclic AMP, is not able to effectively inhibit the kinase subunit. So then the kinase subunit starts to phosphorylate substrates. And the phosphorylation is shown here by this yellow starburst. So the net effect of this molecular mechanism is that when you increase the cyclic AMP concentration, the activity of pKa starts to go up. And it's a relatively concerted transition because these two molecules of cyclic AMP that bind domains A and B are actually cooperating with each other. So that's also a form of allostery that we are studying. And that's why pKa is also called the cyclic AMP dependent kinase. Okay, so what we want to do is we want to modify cyclic AMP in such a way that as we increase the concentration of this modified cyclic AMP, we don't increase pKa activity, but we keep the kinase inhibited, even if we are binding to the regulatory subunit. So an effective way to do that is probably the best way so far known to do that is by replacing this equatorial exocyclic oxygen in cyclic AMP with the sulfur. So if you think about the periodic table of the elements, sulfur is really below the oxygen. So this is what we call an Isolobar replacement. So we're not really changing that much in terms of the valence electrons. We are just replacing a single atom with the bulkier atoms and with more electrons in it. So it's probably more polarizable. So we are changing steric interactions. We are changing the polarizability of ionic interactions. So it seems like a relatively subtle modification from the chemical point of view, but from the functional point of view, in terms of kinase activation, it has a huge effect. And let's see here its effect. So when you increase the concentration of this modified cyclic nucleotide, you see, we call this RP. The kinase activity stays pretty much at baseline. And there's a positive control. They also introduced the cyclic MP to show that you actually have the activation, and the other distereoisomer with the sulfur in the actual position that is instead, rather than being an antagonist, is an agonist. So you see the kinase activity goes up to 100%, the maximal kinase activation. So, but the interesting observation that was known almost 30 years ago, but not explained until recently, is that this analog that we call RP is an effective antagonist for pKa only in the presence of excess magnesium ATP, which is usually the K in the cell. There is quite a bit of magnesium ATP millimolar concentrations. However, if the magnesium ATP concentration goes down, RP switches from an antagonist to an agonist. As you see, if the magnesium ATP concentration goes down, then as you increase the concentration of RP, the activation of the kinase starts to occur. The kinase activity goes up as you increase the concentration of RP. So we found this observation extremely interesting. It basically proved that allosteric pluripotency was at play also for pKa, and was controlled by a very important metabolite, magnesium ATP. So we really wanted to understand it. So first step when you try to understand something in structural biology is try to solve the structure. And that's what our collaborators did. But to our surprise, when they compared the structure of the regulatory subunit of pKa bound to cyclic MP to the same structure but bound to RP, they didn't find any meaningful difference. The difference was below 0.5 angstrom. A root mean square deviation between two structures of 0.5 angstrom is basically of the order of what you would expect because of thermal fluctuations. So that's not a significant difference. And you can also appreciate that if you compare the two ribbons here, the gray, so the RP bound, and the green, the cyclic MP bound, these two structures are really, really similar. In spite of their very different activities, we know that for cyclic MP is an activator, whereas RP in the presence of magnesium ATP is actually an inhibitor. So how could it be that we have similar structures, but different function? So we hypothesize that perhaps the key to solve this paradox is looking at dynamics. So basically, we wanted to understand what is the molecular mechanism for this agonist-to-antagonist switch of pKa. So our hypothesis was that dynamics was the key in explaining this switch. So we put together an experimental design that we called thermodynamics with a capital D to emphasize the importance of dynamics in the thermodynamic understanding of regulation of kinases. So I won't go into details, but I just want to emphasize that there are three pillars to this experimental design. One pillar is a form of spectroscopy that we call NMR, or nuclear magnetic resonance, that you may have seen in some of your courses. And NMR is very useful not only to study the covalent structure of small molecules, so to support organic chemistry or synthetic efforts in general, but it's also extremely useful to map the dynamics to see how structures move in solution at atomic resolution. So that's very useful to come up with atomistic models. Then we try to translate what we observe through NMR into what we called ensemble allosteric models. So we try to use NMR to identify all the different conformations that a given protein can sample. And then we try to use NMR also to map the free energy landscape within which these conformations are distributed. Once we have this ensemble allosteric model, we can make quantitative predictions about what we should expect from enzymatic kinase assays. And then we actually go and do these kinase assays, and we compare what we observe to our predictions. And this comparison is usually quite instructive, and we can learn a lot from it. And based on what we learn, we design mutations of the protein or variations of the allosteric modulators, and we go through a new iteration of this experimental design. So without getting in further details about this experimental design, I'll just tell you what you get if you do all the work. So what you get is essentially a map of what are the conformations accessible to these dynamic proteins. And then you report these conformations, or what we also call states, in a free energy hierarchy. So this G is the Gibbs free energy. So it basically tells you which structure is the most stable. So if you are very low in the Gibbs free energy, a structure will be very stable, will have a very high population. So in the first observation that you have here is that the most stable structure, so with the lowest Gibbs free energy, is this one. And if you look at this structure here of the R subunit bound to the RP antagonist, is actually very similar to the structure that we had seen before. So this actually explains why, when you solve the crystal structures of this complex here, you find the same structure that you would see in the presence of a cyclic MP, although cyclic MP and RP behave very differently in enzymatic assays. And the reason why they do is because their excited states are actually very different depending on whether you have cyclic MP or RP. If you have RP, you have several states with these red triangles that indicate the presence of inhibitory conformations. So conformations that are able to bind the kinase subunit in the presence of magnesium ATP. So if magnesium ATP is able to stabilize these complexes between the regulatory subunit and the C subunit more than the differential between the ground and the excited states, then the net outcome is that in the presence of magnesium ATP, the most stable structures are these complexes between the kinase domain and the regulatory subunit. So essentially, we have inhibition of pKa, and that's why we have this antagonistic response. However, if you were to repeat the same analysis of free energies in the absence of excess magnesium ATP, now we know that the affinity of the kinase domain for the regulatory domain is way lower, because as you remember, the magnesium ATP works as a glue to keep the regulatory and C subunit together. So in the absence of excess magnesium ATP, this complex here will not be very stable and will not be stable enough to become the ground state. So the ground state will still be the same structure that we had before adding the C subunit in the absence of excess magnesium ATP. And this structure here is non-inhibitory. So we have kinase activation. So in this case, RP, apart from the lower affinity, behaves similar to the other agonist that is able to activate the kinase. So with this simple model, and basically realizing that it's essential to look at these excited states, these inhibitory excited states, we were able to explain allosteric pluripotency in pKa. And this was good because it allowed us to identify also other drivers of allosteric switches of allosteric pluripotency. For example, substrate affinities, or what we call, to some extent, Km value from the Michaelis-Menten analysis. So we argue that if we have a substrate with a higher affinity for the kinase, we will essentially have an effect comparable to low magnesium ATP, because the substrate compete with the regulatory subunit. And then we work this out quantitatively to the ensemble allosteric models, and our kinase assays actually confirm this prediction. So we were quite excited about this, and we published this a couple of years ago. But the interesting thing is that similar concepts in general terms started to emerge also for other kinases, such as the Plasmodium falciparum protein kinase G. So Plasmodium is the main pathogen causing malaria, and the protein kinase that is controlled by cyclic GMP in Plasmodium is actually a fairly good drug target for malaria. Now, the difference between PKG and PKA is that the catalytic domain is actually part of the same polypeptide chain as the regulatory domain. As you remember, for PKA, the catalytic subunit and regulatory subunit were actually separate, were two separate polypeptide chains. Whereas here, everything is in a single polypeptide chain. So the regulation occurs through a closed to open transition of this kinase. So in the absence of the GMP, you have an inhibitory closed topology, in which the regulatory domain closes over the catalytic domain, so substrate cannot enter the active site between the small and large lobe of the kinase. And we have an equilibrium between these inactive closed species and the active open species. When you add cyclic GMP, we know that cyclic GMP is selective for the active conformation, so you shift the equilibria towards the active conformations. So this is probably one of the simplest ways to understand allostery in thermodynamical terms through a very simple thermodynamic box that arises from the coupling of two equilibria, this inhibitory two-state equilibrium and the binding equilibrium of the allosteric effector that in this specific case is cyclic GMP. Now, most of the time when you solve structures through crystallography or other methods, you look at the apo-inactive and the holoactive structures, because those are the two most populated structures, whereas the apoactive or the holoinactive states are not very much populated, much more difficult to study experimentally, and yet they are still very important. For example, the holoinactive state is very important to inspire the design, to guide the design of allosteric inhibitors, because it shows how a molecule can actually bind the major state of the apoprotein without causing further activation. So there is a lot of interest in trying to understand this all-inactive state. So in fact, we wanted to understand the mechanism of action of several cyclic GMP analogs known to work either as allosteric inhibitors or partial agonists for this Plasmodium falciparum PKG. And our hypothesis is that they acted by reverting this equilibrium from the active to the inactive states. But we didn't know if they were reverting to the original inactive state or they were stabilizing different inactive states that were not different states from the active that are not able to inhibit the kinase. So again, we use our three-pronged experimental design, thermodynamics with a capital D, and we saw that actually the simplest model to explain the allosteric inhibition, this malaria kinase, was a three-state model in which these allosteric inhibitors, they don't shift completely the equilibrium from the active to the inactive state, but rather they stabilize an intermediate that is quite similar to the active state with one exception. This C-terminal lead that closes over the cyclic nucleotide remains disengaged. And that's enough to actually basically ensure that we still have inhibition. And this is very important because the fact that we stabilized this mixed intermediate as opposed to go all the way to the inactive state allows us to obtain this allosteric inhibition while preserving very high affinity. So as you can see here, cyclic GMP binds in the tens of nanomolar, and 8-NBD, which is one of the best allosteric inhibitors, binds with very comparable affinity. And the ability to allosterically inhibit a kinase without losing substantially affinity is because in this case, we stabilize an intermediate as opposed to the fully inactive state. So now we have seen two stories of allosteric inhibition of pKa and plasmodium PKG. Let's try to compare and contrast these two models. So what's common between the two scenarios? In both cases, central to the mechanism of action were these mixed intermediates in which we had the elements of both active and inactive structures. You know, if you want to use an analogy inspired by Greek mythology, you could say the inactive structure it's a man, and the active structure corresponds to a horse, and these intermediates are a kind of a mixture of the two. You get these centers. So part of the structure of these intermediates will be similar to the inactive, and part will be similar to the active. But the interesting thing is that, sorry, my computer didn't crash, but I think there was a large file, and it took a while to load, and then inadvertently I went to the end. So let me share this, just a sec, almost there. Can you guys see it?
Yes.
So we said basically the emergent theme is the stabilization by allosteric modulators of these intermediates that are called like centorial intermediates, or more scientific terms, in mixed intermediates, because they combine within a single molecule elements of both inactive and active states. And without getting into the details, we mapped multiple other mechanism of actions of allosteric modulators, and we got you know, this recurring motif, these mixed intermediates always emerging. Now, if you think about it, the emergence of these mixed intermediates is that shouldn't be actually too surprising, because often these allosteric inhibitors do contain different moieties, so different parts within the same molecules, that drive allostery of the protein in different directions. So for example, if this is the structure of, excuse me, cyclic GMP, for example, if you keep the base unmodified, so the guanine base, but you modify only the phosphate, you kind of drive the structure, the part of the structure that interacts with the phosphate towards a given active or inactive conformation, whereas the part of the structure that interacts with the base remains as in the active conformation. So if the phosphate drives the structure towards the inactive conformation, then you kind of twist your protein conformation, and you force it to access these higher energy states, what we saw before also in the case of pKa, so those states with higher Gibbs free energy, which are however stabilized by these analogs. So that's why we shouldn't be surprised that the actual state, the actual conformation of the protein targeted by these allosteric modulators is not it does not resemble the structure that we have seen before of the inactive of active conformations, but these mixed intermediates, these centorial intermediates. And there are two key take-home messages here. One, that even when you take two kinases and they have fairly similar active and inactive conformation, so they both look either as horses or humans, when you look at their intermediates, their excited intermediates that are actually targeted by these allosteric modulators, you have much more diversity. So if you just look at their ground states, you will just see all horses or all humans. So you won't see too many differences between different kinases. But if you have techniques that enable you to see, such as NMR, to see these excited states, these intermediates, you will see a lot of differences between these kinases. you will see that although they are all mixed intermediates, they're mixed in different ways. You will see that maybe in one kinase, one excited intermediate has the head of a human and the body of a horse. But you will see that in other kinases, you will have the reverse pattern. You will have the head of a horse and the legs of a human. So you can then exploit this diversity that you have at the level of excited states to selectively target one kinase versus another, when even looking at the ground states, you wouldn't see any hope of selectivity. And this is essentially the reason why we believe that allosteric modulation enables selective kinase inhibition. Now, the other take-home message is that inhibitory states can be obtained usually not in that many different conformations, but active conformations that are not able to inhibit the kinase can adopt many different structures, including structures that are in different phases. And here I had included a part about using phase transitions to understand the mechanism of mutations related to carnic complex, the cancer predisposition or hormonal resistance that I mentioned before. But I see here that I already spoke for 50 minutes. So I think the best use of the remaining time is perhaps summarizing the key conclusions and then opening the panel for discussion. So as we said, basically we saw that through an experimental approach that we call thermodynamics with a capital D, enabled by NMR in combination with simple statistical thermodynamics and molecular dynamics simulations, we are able to map the structural features of these centorial intermediates, for which there is much more diversity as compared to the ground states that are typically inactive or active. And this has a lot of implications in terms of how we can maximize the potency of inhibition, so the horizontal translation of these dose response curves, and also has a lot of implication in terms of allosteric pluripotency, which dictates instead the vertical scaling of these dose response curves. However, allosteric pluripotency has several other major implications. So first, since it shows how the combined action of not just one ligand, for example, cyclic MP, but two ligands, cyclic MP and magnesium ATP, together contribute to the allosteric control of an enzyme, it opens the way to understanding rationally how to combine different drugs. Now, multi-drug combinations can be extremely powerful because you can effectively take advantage of the complementarity between different drugs. But to actually screen for drug combination, it's a major task, because if you have thousands of molecules in one library that you have to screen and thousands of molecules in another library that you have to screen for another site in a given enzyme, the combinatorial complexity is just enormous, and then you don't have enough high throughput typically in a reliable screening method to identify the best multi-drug combination. So you do want to complement blind screening methods with more rational approaches. And what we have seen about allosteric pluripotency offers an opportunity to do that. The other major implication of allosteric pluripotency is that the allosteric drug target is not just the isolated protein in solution, but is the allosteric protein in solution plus the metabolic environment, for example, in our case, magnesium ATP, plus the proteomic context, for example, the given substrate for the kinase, because the substrate itself can act as an allosteric modulator. Okay, so I would like to conclude thanking all my co-workers for the excellent work they did, the funding agencies and my collaborators, and you for your kind attention. I'll be happy to entertain questions if you have any. Thank you.
Thank you so much, Dr. Melanchini. If anyone wants to unmute, to ask a question. Even if you don't want to show your face, you want to mute, or you want to put it into the chat, it's also fine. While we wait, I guess I'll start off with
Yes,
a question that I have.
yes.
Maybe make people more comfortable in asking questions.
You break. We need an icebreaker.
Yes. So for the mixed intermediates, would the goal to create mixed intermediates be to obviously to modulate the amount of dose you need to actually get a response, but would also creating an intermediate cause you to have the ability to prevent the enzyme from actually having a certain function?
Yes, definitely, because I mean, you have to look at these mixed intermediates, but often these mixed intermediates have enough inhibitory capabilities. It all depends on how the different features or the active and inactive structures are distributed within that molecule. So if you preserve enough features that are inhibitory, then the mixed intermediate will have a net inhibitory effect. So if you have a molecule that stabilizes that, then you have allosteric inhibition. If the mixed intermediates as enough moieties that resemble the active conformation, then most likely when you have a molecule that stabilizes that, you get partial agonism. So you can go both ways depending on the nature of these mixed intermediates.
Okay,
So
that makes
that's why
sense.
it's important to characterize them.
Oh, so that gives you more of a analog control on the enzyme's behavior rather than complete inhibition one way or complete
Yes.
agonism, antagonism the
Yeah,
other way.
that's a nice way to explain it. Yes.
Okay, that makes sense.
Or it makes it less binary. Although you may not have a full continuum between the active and inactive structures, at least it's less on-off. You have more subtleties in between these two extremes.
Does anyone have any questions? No, don't feel shy. Just know you have a one-on-one chance right now to ask a question. If not, you'll have to email a question later on.
Yeah, feel free to contact me. Which universities are you in?
I'm at McMaster University.
Biochemistry?
Yes. Yep.
Great.
I guess then I'll drop your email into
Sure.
the chat. Contact. For anyone who might be interested. Oh, so we have someone in biology and
Oh, wonderful. PMB. Excellent. So maybe we'll bump into each other as classes start.
We have a math and biology student.
Oh, excellent.
Perfect for statistical analysis, I guess.
Yes, yes. One of the methods we developed to map allostery is called chemical shift covariance analysis. So chemical shift are basically just some measurements that are very common and very robust through NMR. And then we use covariance analysis to actually see how these molecules concertedly respond to the allosteric inhibitors. That's wonderful that we had people from also biology, PMB, and math. I mean, biochemistry is great. It's kind of, I'm also in the biochemistry department, so that's, we are like at home. Do you teach any undergraduate? Thank you for your question, Hafsa. So usually, yes, in the past, I used to teach quite a bit, but as I got older, I got involved more into administration. So then I had less time for teaching. So actually, currently, I'm not teaching at all. I'm serving as a acting chair of our department, chemistry and chemical biology. So I don't have much time to teach, but in the future, I actually look forward to go back to teaching at least one course. So I used to teach for many years the inquiry in chemical biology course, which is second year for chemical biologists, open to others as well, with Dr. Berti. So that course was a lot of fun. I also taught several graduate courses in chemical biology and NMR spectroscopy. I taught physical chemistry for life science students. Not the favorite course of life science students, I should say, but some of them liked it, not everybody.
Seems like we had a question from Constantine, I'm pretty sure.
Okay.
Oh, right. So first of all, thank you for your talk. Thank you for taking the time to join us here.
You're most welcome.
I am in math and bio, and I've only very recently started to see what other stuff is out there, trying to see what other research is going on in other domains of science.
Yeah.
Something that caught my attention, and I see it coming up in conversation with friends, and I also see it coming up with some relatives that started getting really into health and fitness, but magnesium. I hear from one front that, magnesium, like any other mineral, you need it in moderation. But then I hear from relatives where it's like, oh no, magnesium is the one thing that everybody has a deficiency of, and you should be taking more of that and whatnot. And I saw that one of the graphs in there talked about magnesium ATP.
Yeah.
You had a molecule that was an agonist of pKa,
Yeah.
but then in the presence of pKa was an antagonist, right?
Yeah.
And my question with regards to that is, what exactly, like, what role does magnesium play in there? And is it all the hype that somebody might say out there, right?
That's a very good question. I mean, the role of magnesium is actually very complex. The reason why I talked about magnesium is because usually ATP coordinates magnesium. So magnesium binds ATP quite well. So in fact, usually they come together. Often biochemists usually talk about magnesium ATP as opposed to ATP. And there is a lot of ATP in the cell. It can go from 2 millimolar to 10 millimolar, so very high concentration. And that's because magnesium ATP is used for so many different purposes. So traditionally, magnesium ATP was just considered major source of energy in mitochondria. But then the signaling came, they realized, no, it's not only energy, it's also used for signaling through kinases that transfer the phosphate. And then more recently, there was actually another paper a couple of years ago or so. They discovered that actually magnesium ATP is also very important to keep proteins in solution. So proteins tend to, especially when you get older, they tend to unfold a little bit, to lose their structure, and then they tend to come together and form assemblies. These assemblies could be in the form of what is called liquid-liquid phase separation, or they could form amyloids, so really insoluble aggregates that usually, for the most part, are not functional. So they can actually be dangerous because they are in equilibrium with oligomers that can be cytotoxic. There are some amyloids that can be functional, but it's more of an exception. So magnesium ATP has a lot of different functions. So understanding the effect of magnesium is quite complex. And then there are also magnesium that are independent of ATP. So I don't have a good answer for you or your relatives about the ideal dose of magnesium in a balanced diet, but it's definitely quite important.
Mm-hmm. So just to recap, energy, right? Magnesium ATP, that's the going all the way back to high school biology.
Yeah.
signaling as well. And the third
Yeah.
one, I forget the term that you use.
It's the technical term is that magnesium ATP is an hydrotrope. So hydrotrope is in chemistry is the technical name that we use for small molecules that are able to solubilize other molecules that would not be able to be solubilized otherwise. And in particular, hydrotropes do that without causing the formation of micelles. Because if you think about it, if you take a surfactant, for example, when you wash your hands with soap, basically that's a surfactant. So it forms small bubbles that take the dirt, the greasy dirt inside them, and then you're able to wash it away. So the hydrotrope is able to solubilize greasy stuff, including proteins that get unfolded without forming those micelles, those bubbles, just through interactions that are actually not very well understood. So in fact, we have another project that is looking at the interaction of magnesium ATP with intrinsically disordered proteins that usually would tend to aggregate, such as, for example, synuclein, which is linked to Parkinson's disease. We are trying to see how it interacts with magnesium ATP to see how magnesium ATP interferes with the amyloid formation by synuclein. So that's the hydrotropic effect of magnesium ATP.
Yeah,
It was a science paper a couple of years ago, I think.
Yeah, I looked it up right now. I think it's
You found it.
yeah, a paper by Meow from 2021. Yeah. Okay, well, thank you so much for that. Very informative.
Thank you, Constantine.
Thank you so much again, Dr.
And I saw
Melanchini.
some smoke coming out in the screen of Israel. Hopefully it's not your laptop.
It's not my laptop. I don't think, maybe I'd have to check for some other sources. Hopefully something bad. Thank you so much again, Dr. Melanchini, for giving this presentation. I hope if anyone has any questions, of course, I hope they can email you and ask away. And
Thank you so much.
Thank you everyone for attending.
Thank you so much for organizing this. I did try to make it more accessible for a wider audience, just reviewing more like basic concepts, but I'm aware of how technical things get quite quickly. So don't hesitate to contact me later on if things are not clear.